গুড মর্নিং ম্যাডাম। শোনা যাচ্ছে?
হ্যাঁ, শোনা যাচ্ছে, শোনা যাচ্ছে।
আচ্ছা, ঠিক আছে।
ঠিক আছে, শুনতে পাচ্ছো? আবার বেরিয়ে গেল।
হ্যাঁ স্যার, আপনার কথা শোনা যাচ্ছে,
ঠিক আছে, ঠিক আছে।
ভালোভাবে শোনা যাচ্ছে।
Hello, can you hear me? Good
Yes sir,
morning.
you are audible, you are audible.
Hello, everybody.
Hello?
Hello, can
Hello?
you hear me? Yeah,
Yeah, audible, sir.
okay, thank you. Good morning, Chandanda.
Good morning.
Oh, nice to see you.
Thank you.
Hello, Dr. Anjan Sengupta, can you hear me? Hello.
Good morning, Professor Chakraborty.
Good morning, a very good morning. Welcome.
Sustainability is treating ourselves and our environments as if we are to live on this earth forever. Aaron Wood. A very good morning to everyone here. I take this privilege to warmly welcome all our delegates in today's national webinar on challenges in environmental microbiology. To welcome is to show honor. To welcome is to show integrity. So now I am inviting the Honorable Principal Madam of Chagda College, Dr. Shagota Das Mohanto, to deliver the welcome address. Please, Madam.
শোনা যাচ্ছে অনন্য?
ম্যাডাম একটু জোরে বলতে হবে।
হ্যালো, শোনা যাচ্ছে?
হ্যাঁ,
হ্যাঁ,
শোনা
audible.
যাচ্ছে madam।
শোনা যাচ্ছে।
শোনা যাচ্ছে।
ঠিক আছে।
শোনা যাচ্ছে।
ভাইস চ্যান্সেলর স্যার জয়েন করেছেন তো? অনন্যা,
আমি তো দেখতে পাইনি এখন। ম্যাডাম আমি দেখার চেষ্টা করছি, কিন্তু এখনো মনে হয় জয়েন করেননি।
না কিন্তু সেটা একটু হ্যাঁ হ্যাঁ স্যার আছে।
Yes,
স্যার হ্যাঁ ঠিক আছে। হ্যাঁ স্যার আছেন। ঠিক আছে।
good morning,
হ্যাঁ,
good
sir,
morning.
Good morning. Good morning.
Good morning, sir.
Namaskar. Namaskar.
Namaskar.
Very good morning to all of you who are present here. আজকে চাকদাহ কলেজের ডিপার্টমেন্ট অফ বটানি এবং ইন্টারনাল কোয়ালিটি অ্যাসুরেন্স সেল মিলিতভাবে ন্যাশনাল লেভেল ওয়েবিনার করছে। অন্যবারের মতোই এটা আমাদের জন্য খুব সম্মানের এবং মর্যাদার যে আমাদের অভিভাবক তুল্য আমাদের লিডার, দক্ষ প্রশাসক, কল্যাণী বিশ্ববিদ্যালয়ের উপাচার্য অনারেবল প্রফেসর ডক্টর শঙ্কর কুমার ঘোষ মহাশয় আমাদের সঙ্গে আছেন। আজকে অত্যন্ত বিজি শিডিউল তার, তবু আমাদের সময় দিয়েছেন, সেই জন্য আমরা আন্তরিকভাবে তাকে কৃতজ্ঞতা জানাচ্ছি। আমি সেই জন্য I must not take much time. I would like to welcome Honorable Vice Chancellor Sir. Then I would like to welcome Dr. Chandan Sengupta, former Professor, University of Kolani, Department of Botany. I would like to welcome Dr. Ranudhir Chakraborty, Senior Professor, Department of Biotechnology, North Bengal University. All of you welcome, sir. I would like to welcome Dr. Bernard F. Rodriguez, Professor, Department of Botany, Goa University. All of you most welcome, sir. You are with us today, and we must. Your lecture will enrich us. I would like to welcome আমার কলেজের সমস্ত অধ্যাপক অধ্যাপিকা শিক্ষাকর্মী এবং স্টুডেন্টদের, আর অন্য যারা পার্টিসিপেন্টস আছেন অন্য প্রভিন্স থেকে, দেশের বাইরে থেকে, তাদের সকলকে স্বাগত জানাচ্ছি। অনুষ্ঠান খুব সুন্দরভাবে সম্পন্ন হোক এটাই চাইবো। All of you most welcome. Thank you.
Thank you, Honorable Principal Madam, for her warm welcome address to all of us. It is indeed my honor to stand before you to introduce the chief guest of this webinar, the Honorable Vice Chancellor of University of Kolani, Professor Dr. Shankar Kumar Ghosh. Dr. Colonel Shankar Kumar Ghosh is an eminent scholar and academician in the field of biotechnology. The area of his research work includes genomics, cancer immunology, DNA barcoding, bioinformatics, medical and animal biotechnology. He is an outstanding academic achiever and administrator with lots of experience. Now I am requesting sir to give the inaugural address. Please, sir.
Good morning, all of you. Very good morning and welcome all to the participant and others on behalf of Chagda College as well as the Kolani University also, because the Chagda College is one of the most vibrant colleges under Kolani University, and they are doing a very good job during the COVID period. And the students of the Chagda College are one of the prominent students in our university, where they want to stand every year and every time. It's a really great pleasure for me to have a with this webinar today. Firstly, I congratulate the principal ma'am, Shagota ma'am, who invited me to a part of this webinar. And when he discussed this issue with me, I I told her that "Arey, one of my PhD fellow, Randeep Chakraborty, is with you, and he's a very brilliant guy during my PhD program at Bose Institute. I welcome Ranadir if you are here, and
Yeah,
also
thank you, Professor
uh,
Ghosh.
yeah, thank you, and our esteemed professor, Professor Sengupta, Chandan Sengupta is a really renowned professor in our university, just retired. However, he is closely associated with the university. Hopefully, I have a very good relation with him and extremely happy to see today's program. Thank you. And other uh, mm, international expert and the student and the research scholar, all of them are very welcome to this part. And I just want to share because the botany department, the alumna has tried to organize this kind of program. It's a really excellent program. I hope there are lots of people, lots of research scholars will be benefited out of that. And what I want to just sharing with you, we want to enter into the new normal life. All of you know. So every day there are very good webinar, and now we have accustomed ourselves, or we have to make mindset ourselves to go the webinar. I have five webinar today, basically. So I try to make presence in every time because we need to go this program in near future. I don't know how when we are again meet with the lots of students to take the classes. Very difficult, in my opinion, is very difficult. However, in the present situation, what we can do, because we are waiting for vaccine, right? And COVID-19, Ranadir will and Professor Sengupta also will tell the COVID-19, their activity, their genomics, their migration, everything, because it lies on the microbiology. And so what basically want to address that we are in the new normal. That's true. And now the vaccine, we are waiting for vaccine, and when the vaccine will come, and how long it will exist, and when the immunity will come, and when the booster will be given, and different kind, because it is a RNA virus. All of you know RNA virus vaccine is better than vaccine, DNA virus vaccine, but it is a very big, more than 30 kb RNA, and there are lots of different strains of these strains, or haplogroup or haplotype, whatever may be, and there are. So it come from the Wuhan. Definitely, it comes from the China, and it goes in different part. Just I am focusing some news on the Facebook, because Facebook, that is the most important migratory news. Facebook is the most important migratory news, and how the viruses has migrated from Wuhan to other places. A publication came in the Facebook, so Facebook report that gives a very nice deliberation how it comes over there in there are lots of millions of data that they have analyzed, and how it comes. This is the one way. And another way, the nucleotide mutations and other part, this is also important. People, scientists are working. There are thousands of genome sequences already have done. Even in India, all of you know, the NIBMG, Indian Institute of Science, and other institutes have created a very a positive role for sequencing of the whole genome sequence of these COVID-19 viruses. So it's a good approach from our side. And India also have spent more than 100 crores of money to develop the vaccines. There are 15 different laboratories. They are working on that. Out of that, three vaccines are, all of you know, three vaccines are very good shape, and they are on the They are on the different phases of trial, and might be one or two maybe very give positive result, and we will get the vaccines near future. This is the our idea. Now, what is my point? My point is that during this period, the very important point is the how we can proceed, not in 1 year, not in 2 year, up whether the vaccines will effective 100% or not. There might be some question. There is any no doubt of it. Those who are preparing the vaccine, they will raise this question. Okay, because the vaccines, when the booster will come, the first booster and first injection, then when we want to go for the second booster, Moderna, Moderna vaccines, the RNA vaccines has created some of my fellows, and we are discussing this issue. And really, they have also a very big question in their own mind. Anyway, but right now, my feelings right now, who we have to go for some kind of very important issues that we know every time, because this COVID-19 and the situation came last 6 months. So we have a everybody have a idea of the 6 months idea. 6 months. So we are basically newborn baby regarding this COVID-19 and pandemic and their part. So it is proved, or everybody knows, that we want to go some kind of guideline. So who guideline, ICMR guideline, or a government of different state government guideline, and they guideline are clear. And these guideline basically are the social vaccines. So we have to change our behavior. We have to think our positiveness in such a way that we may do some kind of vaccine. So behavioral vaccines or social vaccine, we want to term is a social vaccine. These social vaccine, the social vaccine, that first social vaccine is lockdown. So we faced lots of lockdown, and still it is continue, and today is also lockdown. So initially it started lockdown nationwide, and because lockdown we want to make separate to each other, and we have to prepare ourselves, to prepare the masks, to prepare the hand sanitizer, to prepare the other instrumental part. So lockdown is one part, and second part is the marks. The marks, wear of marks is very important in this COVID situation or COVID. All of you know, when a cough or sneeze, that more than 3,000 droplets are coming, and the speed is more than 50 miles per hour. So you can see when we go for the one sneeze or cough, there are lots of droplets are coming in your area. So that in a locality, if you go without masks, you don't know where the droplets is moving around you, because droplets are coming from cough or sneeze. So that might be in around you. You can tell that no need for marks because I am very strong, and you are entering in the market, or you are entering, or you are walking in the road, or you are going for bank. So you have no idea the droplets around you or not. So that's why your protection is your protection. That you wear masks is very nicely. In our body we have a six odd natural orifices. There is a orifices of mouth, there is a orifice of nose, there is a orifice of eye, and there is a orifice of ear, and orifice of nipple, and the vaginal organs or the rectum. So it was found that only three natural orifices, mouth, nose, and eye, is a predominant risk factor to enter the viruses in our body. And only one report has come up in the rectum. So far is scientific attitude. So these three are most important, that we need to make ourselves away from these droplets particularly. That means the virus is a droplet. So you want to make the shield also. Why? The droplet cannot enter. So if you go in any places, apart from marks, you can enter, you can take the shield. The eye will be protected, and also you can take the specs. Definitely, you should take spec along with the marks, and marks should be. There is no need for N95 marks. People are hungering for N95 marks with a valve. It is no necessary basically. If you go in the hospital, or if you go in the risk areas of this COVID, where you can see the COVID patients, so many, you can wear the marks of N95. But double marks or surgical marks is a very sufficient to make. But you have to wear the marks in a public place. I am in home right now with my family. I no need for marks because we are every time along with. So there is a no question of transmitting of theirs, because if I have a part on that, so my family will be affected. So in that way, why the marks is very important. Now, secondly, the hand sanitization. Why we want to go for the hand sanitization? That is very important. Whether the frequently we want to do, or there is no need for, because if you want to night sleep, you want to wake up on the 7 or 8 a.m. So entire night there is no need for hand sanitization, right? But even though if you want to go for in the market or in the risk factor where the virus droplet can come, and that risk you have to identify in yourself. Somebody are asking the question, how many times we have go for hand sanitization? It is not a good question in my opinion, because you have to, like a diabetic, if somebody has diabetes, he should know we do not take the sugar, rasgulla and others, because we know that might be create our problem in the rising the sugar in our body. Similarly, now we have to we already learn how the virus is spread from one place to another places, the droplet. So the hand sanitization is also a correlated the risk factor, that where you want to enter and come, like if you go for the withdraw of money from ATM, just if you go for 5 ATM, so you need to 5 times hand sanitizer, because this touch of this part might be risk for you. So you need to do the hand. If you go in any flathead in Bengal, in particular in Calcutta, every flathead have a COVID positive. Every flat, 10th year flat, you just take a note on that. Every flat there is a or fourth or seventh. Just in the morning, I came to know one of my professor that he is in third floor, and fifth floor there is a positive. 11th floor there is a positive. And we don't know how many positives in this flat. So it's very important to make the hand sanitization in that cases, like a where the other person or touch on any part, that might be the some kind of droplets which may create some problem or get in part. And third, the social distancing. All of you, the distancing is the most important social vaccine, that you have to distance to each other, like the minimum 6 feet is an important issue. The question is whether we can make this social distancing, how long we can go. In my feelings, you can go very big time. And it was history told us that if you want to infect in this kind of pandemic, and again it will come after 2 years, 3 years, we do not have any idea about this COVID-19. So as it is a very big RNA viruses, and the less mutations are there, it was proved. So this is the positive sign for us only. Otherwise, I have how many, as because there is a no proofreading capacity of the viruses, then there might be different kinds of variation or genotype may occur. The HPV, all of you know, because in microbiology, there are 142, there are different types of HPV. and only 5 type is a risk factor, and 16 and 18 is a predominant in the cancer. In the cancer, if you go in the whole genome sequences, and part of that, there is a lots of information, genetic, epigenetic of the HPV, and which is related to the cervical cancer. So these are the long term. Even if you go the Epstein-Barr viruses, which causes the nasopharyngeal cancer, viruses that causes the nasopharyngeal cancer. We do not have that much genotype of these Epstein-Barr viruses, but it was found the 99.99% having the Epstein-Barr viruses in their genome. So I have a Epstein-Barr virus, and somebody also have a Epstein-Barr viruses genome in their genome sequences, and lots of viruses are in the genome sequences in our body, lots of viruses, and we do not have any idea whether these COVID viruses can integrate it, or any portion of the virus particles can integrate it into the in a human genome, and that is transmitted throughout the body. So the migration of these COVID viruses is still in question. Where it comes from the pangolin or bat. There are lots of evidence also there, how it migrated from one place to another places, and genomics people are trying to do this kind of work to get the different genotypes in India, in China, in Brazil, in Australia. There are lots of people are there. So that might be helpful to create some kind of conclusion. But in my opinion, it's very difficult to say the rigid conclusion right now. We are giving lots of information to everybody, and on the other hand, the drug, there are lots of drug are treating in the COVID. There are lots of drug are treating in the different diseases. That drug also help us to treat the COVID patient. There are 5 or 6 drugs is available, and the drug is already FDA or approved drug. That means already there are no negative effect into the body, but that drug also can combat the COVID-19. And right now, artificial intelligence has come up, big data analysis has come up. So scientists have developed this kind of drug, and pathways. We are well known of the pathways of the drug effect on that, and there are 8 or 9 different kinds of drugs has now available to combat the COVID situation. So hopefully all are in conclusion that we need to go for the new life, and we have to develop our mindset in such a way that this may continue how long. We cannot, nobody can say right now. Everybody can tell, but it is a confusion, and there is no rigid data, and there is no rigid information. There is no good rigid publication that we want to finish these things in the March. And if the vaccine will come in December, again you can see there are lots of debate, because a classical vaccines, it needs, somebody is asking, this vaccine can last for 3 years or 2 years, So if you want to put this vaccine and you have to wait 3 years, then you can tell it will continue for 3 years, right? So classical process of this production of the vaccines and this kind is very, but we have to face our problem. We might be very positive, so that we can give some kind of good boost up to all of them. And in human being, right now, we can tell the world is a one family to combat the COVID-19 in the area of environmental microbiology. I hope everybody will enjoy today's program. I have another meeting in 11:30 with the DBT, so I may leave. And if I want to near future, I definitely I will listen your talk through YouTube, and Professor Sengupta as well as my friend Ranodhir. With this few word, once again, I thanks everybody. Thank you.
Thank you, sir, for your valuable speech. It is immense pleasure to have you as chief guest in this webinar organized by Botany Department, Chakda College. Thank you, sir. Now it is the time to introduce the guest of honor, Dr. Chandan Sengupta, retired professor, Department of Botany, University of Kullani. As a microbiologist, he brought a great change in the field of soil microbiology, particularly using PGPR. He has published nearly about 120 papers in different national and international journals. 28 PhD scholars are awarded, and one scholar has submitted her thesis. I am extremely grateful to him for introducing me to a relatively new, flourishing arena of soil microbiology. To me, he is a perfect example of great philosopher, guide, father like figure, and definitely the pathfinder of my research work. So now I would like to request Sir to deliver the keynote address of today's webinar. Please, Sir.
Thank you, Ananda. A nice introduction. শুনতে পাচ্ছি সব? শোনা যাচ্ছে?
Yeah, audible, sir.
Okay, thank you. Thank you, Ananda, for your nice introduction. Good morning, everyone. Although I am very much sure about it, that last 5.5 months, we are not in good condition, both mentally as well as to some extent physically, due to the deleterious effect of coronavirus, which not only posted us in the pandemic situation, but as Honorable Vice Chancellor sir mentioned, that we don't know when we may actually escape from this devastating effect, from the deleterious effect. Anyway, as the seminar is actually based on environmental microbiology, so I restricted my keynote address on this particular topic. Frankly speaking, coronavirus is not the only virus which is actually responsible for our deleterious. There are large numbers of other viruses, bacteria, microfungi, fungal spore, even to some extent some protozoa also, all belonging to the group microbiology. They continuously hosted various types of problems and diseases. And for this, we people are very much afraid about this. And consider it is actually a great challenge to the microbiologists. How we will cope up the problem, how we tackle the problem, how we will accept this challenge. And in that respect, the focal theme, the challenges of microbiology, I think it is a very justified and appreciated approach to the organizing committee for choosing this subject as the focal theme of this seminar. And for this, I express my deep gratitude and thanks to the organizing committee, particularly head of the institute, Principal Madam, Dr. Shabata Das Mohanto, for inviting me in this seminar and give me an opportunity to deliver the keynote address in this auspicious occasion. Thank you, madam. Now, let it be starting some other ways. Long back, when
Sir, little bit interruption. Sir, please unmute your video.
video, that means
Sir, please unmute your video.
নিচে দেখবেন একটা আইকন আছে ভিডিও লেখা, ওটাকে কেটে দিন।
স্যার ভিডিওটা আনমিউট করুন। স্যার শুনতে পাচ্ছেন?
হ্যালো, উনি বোধহয় মাইক্রোফোনটা মিউট করে দিয়েছেন।
আচ্ছা আচ্ছা আমি একটু
আপনি ওনাকে
স্যার হ্যালো
ফোন করুন।
হ্যাঁ হ্যাঁ সেটাই করছি ঠিক আছে
হ্যাঁ ঠিক আছে এবার হ্যাঁ হ্যাঁ
Sir, you are audible. You can continue.
okay. Now let it continue my lecture. And long back, when the subject microbiology is not in the proper shape, at that time, some pioneer microbiologists, for example, Winograssi, for example, Bezerin, for example, Pasteur, and many others, based on their extraordinary thinking power and monumental ideology about the subject, considered that time to time the microorganisms will more and more in the environment, and each and every time this creates a lot of trouble in our life. For this, we have to cautious. We have to take care about it. Otherwise, it will not possible to us to sustain in this environmental system. In the lecture, I would like to mention many students ask me that how the microorganisms comes on the environment and how it is interact with the environmental factors. I think it is an opportunity for me to tell something in this point. Long back, near about 4.5 billion years ago, the first environment with this environment was created. Although the environmental factors is not like that as exists today. And 0.5 million years after, that means 4 million years ago, the first life form, the first microorganism actually originated on the Earth's surface, which is a very preliminary and primitive and rudimentary types of microorganisms. And from that day to still today, the environment changed a lot, which became sustainable to our living system. And at the same time, microorganisms are also changed a lot through the process of mutation, adaptation, and evolution. And now a variety of types of microorganisms exist in nature that different form various morphological groups, different metabolic groups, and genetics organization. So nowadays, the environmental microbiology, we are actually considered in various fragments, soil microbiology, water microbiology, microbiology, etc. And to some extent, we are also interested about gut flora microbiology. We're interested about the human body surface microbiology. All this is part of environmental microbiology. Now come to details to some extent about the soil microbiology. It is a well-known fact to many of you that there are large numbers of bacteria present on the soil, and they are actually root plant growth promoter. They promote the plant growth in various ways, and which are now it is considered that it is an alternative source of fertilizer and manure. Moreover, it does create any environmental pollution. It retains the soil structure, soil properties, and moreover, it is not cost intensive. Another important factor about the soil organisms, or soil microorganisms particularly, we obtain a large numbers of organisms obtained from soils which are antibiotic producers. Right from 1928, when Alexander Fleming first observed that the growth of some organisms and bacteria is checked by presence in the vicinity of Penicillium notatum. And based on this, after that, Alexander Fleming and Chain and Florey discovered the first antibiotics, penicillium. And these penicillin are actually the first antibiotics, and from that day to today a large number of antibiotics we obtain from the soil, and actually the soil-borne microorganisms which produce these antibiotics. Even today, when you obtain or search newer antibiotics, then always we search the soil that if we get some new organism that will be produce some new types of antibiotics. And today, antibiotic therapy is one of the main pillar of medical science. Like that, we are also interested about the aquatic microorganisms. Aquatic microbiology is another important factor, another important area for teaching and research. We know it actually regulates the growth and development of aquatic organisms, particularly fish, which we consume. So it is another area. And another area is aeromicrobiology. We people are interested about microorganisms in air. For example, we are interested about hospital uh, atmosphere microorganisms of schools, particularly the schools of nursery level, where the children are actually growing. So, considering all the facts, in a roundabout way, we'll find that microbiology in the environment is really a very sound and very proper science, and we have to think in a better, and in a good way. Now, in my personal opinion, as I involved with teaching and research in the subject last about 40 years, I may consider that it is not possible for a particular man, for a particular group of person to do everything in his own hand, in single hand. So we have to take the problem in very sectional way, very fragmented way. How? I just put two examples based on my research work, which is actually at the vicinity of this institute, that is Chanda College, and from which you people realize what is the problem is and how I have to come up with the problem. Few years back, some agricultural men of my district, Nidhanchandra Krishi Vishwavidyalaya, asked me the agricultural field just beside the national highway from Chagda to Ranaghat, which is very close to this college, is not growing the crop properly. Why? As because of the fact the environment is very much polluted with some poisonous gases. What are those gases? Those gases are carbon monoxide, sulfur dioxide, nitrous oxide, etc., and which actually create the stress situation for which the growth and development of the plant is very much affected. Naturally, the yield also reduced. I took the problem and I obtain some bacteria which is already habituated in this atmosphere, particularly from the soil atmosphere, and we introduced in heavy dose of this bacteria in the soil as inoculant, as because of the fact that it are also good plant growth promoter. And we find this radically improve the situation and improve the crop production. And the person which worked with me with this problem, and based on which who actually submitted her thesis, she is Dr. Ananya Rai Chaudhary, one of the organizing member of this seminar today. I put another example from my research work in case of aquatic microbiology. About 10 years back, some professor of chemistry department of my university asked me that there are some villages where the rural people are actually inhabited around the Chagda town. They are actually consume the water from tubewells, shallow tubewell, and the water of this tubewell is heavily polluted with various types of heavy metals, particularly the arsenic. And if we don't remove this arsenic, then actually this create a lot of problem in their life. Not only in their personal life, but when they use this water in irrigation purpose, the crop that also harbor these heavy metals and the growth of these plants are also hampered. For this, we check the situation and we obtain some bacteria from this water surface, which is have the fantastic ability to resist this heavy metal. Then after that 40 fund, we found that after some level of genetic modification of these organisms, when we use these organisms as inoculant, then the condition is partly or more or 100%, not only 100%, about 80 to 90% was good. So this is another point in which exploitation of soil organisms or environmental microorganisms or aquatic organisms for the good growth and development of the human society. And the person who did this work with this aquatic of Chakda district's water, actually, Dr. Ananya Rai, not only Ananya, she is Adita Banerjee, who submitted a thesis based on this work. So I don't put this example just not advocating me. I just put this example to give you an idea, which is a realization that the problem is vast, but we have to choose some problem in sectorial basis, on segmented basis, and that will be very good for all of us or remove the problem. That will be the real challenge which we have to accept, and we have to overcome this problem in very, very national way. I am not going details as we are already run some delay. And so as there is some paucity of time, I just leave before I give two information. First of all, definitely two renowned microbiologists can guarantee, and I am sure about it that from their lecture we will get a lot of knowledge and inform our knowledge. We actually verify our knowledge in the field of environmental microbiology. And secondly, I again thanks to the authority of organizing committee for inviting me and choose this area of the or the focal theme of such webinar, which is a very, very active field of teaching and research nowadays, not only in the field of microbiology, but in the field of human welfare or human growth and development. Thank you.
Thank you, sir, for your words regarding challenges in environmental microbiology. Thank you. The next resource person is the eminent researcher in the field of biotechnology, Professor Dr. Ranodhir Chakraborty. Dr. Ranodhir Chakraborty is a senior professor of Department of Biotechnology, North Bengal University. His main research interest is on environmental microbiology, microbial genetics, and microbial genomics. He published 72 papers in national and international journals. He guided 13 PhD students and 7 are working under his supervision. Now I am requesting sir to start his kind deliberation. Please, sir.
Okay. So I want to share the screen now. The screen is visible to you?
No, sir.
So anybody who is guiding you, so I'm presenting the screen now, right?
Yes. It came, sir.
But the screen is not visible.
Screen is not visible.
You should see my slides now. I need to share the screen.
Sir, you have to put the slides in front of that. Then it can be visible. Just now stop presenting and then again present that slides.
Okay, is it visible now? Is it visible?
Not yet, sir.
Not yet. Okay. Just hold for a few seconds. এটা করি ওকে
কি আছে পাওয়ার পয়েন্টে এটা তো
Yeah. Is it visible now? Hello? Can you see it?
Yes, sir. Visible, sir. Visible, sir.
Yeah. So, before I start, let me just put my regard to Honorable Vice Chancellor Professor Santosh Kumar Ghosh, who has already, given a very nice introduction about the viral genome complexities, about the drugs and awareness. But I'm not going to speak on COVID-19 today. Then let me put my regard to the Principal Dr. Swagata Das Mohanto for beautifully welcoming all the speakers and participants today. The keynote address given by Professor Chandan Sengupta, who is also a senior professor and also my senior in terms of research, for a wonderful background. It is again to give, to bestow my respect to the co-speaker, Professor Bernard Rodriguez of Goa University. A special thanks to Dr. Anjan Sengupta, because we shared the table sometime in Bosch Institute. Beautiful anchoring by Dr. Anunna Rai Chaudhary, the entire team of Chagda College for organizing a national webinar in challenges in environmental microbiology. Now, I need not to say about, the what about am I to speak today? But some challenge is there, and that's a challenge of antibiotic residues and what impact that it has on the environment. What about the microbiome and also the bioremediation strategies? So if you look at this figure, you will be finding that, 10,000, we have a reference point. The reference point is the birth of Jesus Christ. So if you take Jesus Christ's birth as a reference time point, and if you move back to 10,000 years before the birth of Christ, until today, which is 2020, and we are heading towards 2050. If you look at this 500 BC, and then 180 means 1 year after the birth of Christ. And if we move up to 1750, and 1757 is the Battle of Plassey, right? The Industrial Revolution, you will be finding that within 1900 to 2000, there is a massive increase in human population. So basically, one of the questions might be coming into your mind, that what the change that has occurred, that has produced a tremendous impact on demography, of human demography, that the population is on the axis of logarithmic expansion. the reason, if you trace back, there are two principal reasons, right? The life became more beautiful and easier with the advancement of science and technology. And that happened post, and that happened, that increased maximally post Second World War. So 1900 to 2000, we have seen two World War situations. And two World War situation, when we overcome, then there is a plethora of changes everywhere. And one of the most important change that happened in the history of human civilization was the discovery of antibiotics, which just Professor Chandan Sengupta has just told something, some line, which will be my touch line, which will be my cue line, because the development of antibiotics has revolutionized medicine and saved thousands and crores of people's lives. So when you have reduced the mortality, there is an increase in the population. And this is the gentleman, Alexander Fleming, which he spoke about in 1928 with the brilliant observation that this fungus, penicillium, was inhibiting certain bacteria. And from there, his students have isolated the compound, and it was in a huge demand. So when there was a huge demand, there was an industry support to bring out tons and tons of antibiotic. But interestingly, as I said, that antibiotics revolutionized medicine. And when it was introduced in 1950s, it increased the chance of survival. At that time, and even today, many patients die out of pneumonia. And the introduction of penicillin increased the chance of survival from 25% to 80%. And if you look today, the entire modern medicine depends upon the access to effective antibiotics. Now, this, I want to draw the attention to all the listeners and observer of this webinar to this clairvoyance of Fleming. So when he was receiving the Nobel Prize in 1945, he warned about the danger of antimicrobial resistance. He said, just within the quote, the time may come when penicillin can be bought by everyone in the shops. Now you replace the penicillin by antibiotics. And if you ask yourself now that you go to any shop and ask for any antibiotics at any time point, then there is the danger that the ignorant man may easily underdose himself. And if he does underdose, and by exposing his microbes to the non-lethal quantities of the drug, means whatever the small amount of the drug that is present, that will make the entire microflora resistant. So this is the clairvoyance. Someone who could read the future. We call it clairvoyance. And that we will be speaking about, that the amount of, antibiotic being used, not only for the human, but entire population has to be fed. And one of the principal feed is the proteins. And the protein sources are milk and meat. So you require a large farming to be practiced throughout the world. And when you are farming the animals, even the poultry, as well as the fishes, then what you do, you want to grow them healthy. And for doing so, you pump in antibiotic into the feeds. So let me give you an example. Let's say that China, in the livestock industry, they use 15,000 tons of antibiotics are used in the livestock industry. So you can imagine the world's most populous country, and the next is India, and we do not have any idea about about to what degree of antibiotics are given prophylactically to these type of industries. It could be a shrimp industry, it could be, aquatic fish farms, poultry farms, the cattle farms, the pig farms. So we don't have an idea, but I'm sure this will be near to, the consumption of the Chinese people. Now, if you look at this figure, this is something which I would like to correlate with the environment. So this is radioactive forcing component. Now, what is radiative forcing components? It is the difference between the sunlight that enters the Earth's surface, absorbed, and the energy that is reflected back. When you make a difference out of that, that actually influences the climate. So radiative forcing is like that of the climate forcing. And if you look at here, then aerosols are there, very tiny things, okay, which is present massively over ocean, desert, mountain. But interestingly, it may be tiny, but impact is heavy because it leads to albedo effect. What is albedo effect? It leads to cooling of the, of the planet. The planet's reflective capacity is called albedo, right? So aerosols, they contribute to the cooling effect. But if you look at this picture, you'll be finding there are a large number of components which is contribute to the warmth of this planet. Means our planet is being warmed. If you forget about the carbon dioxide, ammonia, nitrogen dioxide, you come here, there is a net anthropogenic component. And in this anthropogenic component, there are several semi-synthetic antibiotics are also contributing to this particular carb. And that also, for certain, this is going to contribute to the global warming. Sorry? Hello? Can you hear me?
Yes, sir, you are audible.
Okay. But I cannot move to the next slide. Let me try it again, right?
Yes, sir.
Please hold, because these are the technical glitches which we are not accustomed to. I'll move closer. Yeah, sorry. Now, let me just reinitiate what I was talking about. So if you look, can you hear me now? Hello?
Yes, sir, you are audible.
Okay. Sorry for this glitch. So you see the trend of antibiotic consumption in India, for example, let's say, it is the data from 2001 to 2015. And these are the color graphs, which shows that, how much of these type of antibiotics we consume every day. And this is the daily defined dose per 1,000. For example, if you make a mathematical calculation, for example, let there be a prescribed dose of an antibiotic per person per day is 1,000 milligram tablet. then the total consumption of that antibiotic in India per day, for example, let's say if you are taking a fluoroquinolone type of antibiotic, it could be norfloxacin or ciprofloxacin, then India per day consumes 1300 kg of fluoroquinolones. So you can just imagine the amount of antibiotic consumption by human beings per day, right? And if you think about who else is supplying this, so you will be finding there are a number of formulation companies that are manufacturing various antibiotics for human use. And here you will get the name of the antibiotics and the number of formulations per company you can get it. Now, which are the leading antibiotic formulation companies? Let me just tell you why have taken this issue, because India and China are the principal producer of antibiotics throughout the world. So the West has stopped producing antibiotics. So India and China are the main producing hubs of this. Amongst India, you will be finding, and these are the principal name of the companies that are producing antibiotics, and the number is 39, means 39 different type of antibiotic formulation. For example, Cipla, when you are considering Cipla, there are 39 formulation that are being produced in India by Cipla, okay? Now, what happens to a very, very, naive visitor or listener to this? We often hear about a term called antibiotic resistance. Is that the antibiotic resistance is the term when, or the issue that has cropped up only after the usages of antibiotic? The answer is no. In a population, given population, if you look at this one, this is a dividing population. There is always in a population, some fraction of the population is having a mutation which can handle the antibiotic at any point of time. So if you look at these white dots and the red dots and these blue dots are the mutants in a population, right? And which are outnumbered by the wild type. So therefore, in due time, this population will increase, and there will be increase in these red dots and also the blue dots and yellow dots. These are the mutants which are always there in an environment. But suppose in that particular environment, it could be your intestine, it could be soil, it could be water, whatever it is, because microbes are present ubiquitously. So if there is a presence of such antibiotics, all these white dots are going to disappear, right? And these small red dots are going to, survive in that particular selective condition. So if you allow, these mutants will grow and divide in the presence of antibiotics, and this is going to spread throughout the environment. My dear ladies and gentlemen, so this is the problem of 21st century menace. One of the worst challenges that the human civilization has to face is the facing of the antibiotic resistance problem.
Dr. Chakraborty,
Yeah.
can you hear me? Sorry for the interruption, but
Yeah.
the visuals are not coming.
You are not getting the visual?
Oh no, the screen is not sharing the visual space.
Okay, let me try it again.
It is humble request to all the participants that please stop presenting your screen. Don't press on the present button, please. Mr. Sabjar Ahmed, please stop presenting your screen. It is humble request to all the participants, please don't press on present button, please. Don't press on present button.
Q.
Hello, can you see the screen now?
Yes, sir.
Okay. Thank you. So can you just give me cue where from you did not see the slide, but you heard it?
Professor Chakraborty, sorry for the interruption. The video is still not visible. The slides are still not visible.
Excuse me, everybody, I'm sorry. Now can you hear me? Hello?
Yes, sir, you are audible.
sir.
Okay. Did you see this slide before?
Yes, sir, your slides are visible that time.
Okay, this one, did you see this slide before? When I was discussing about the trends in antibiotic consumption in India?
Yes, your
Well, let me say.
antibiotic lists are visible.
Please.
Antibiotic lists are visible in the table.
Okay. Now the number of formulation companies, did I say all these things before? That is number of formulations which are manufacturing various antibiotics are presented in it. And this is the leading antibiotic formula and I was talking about the Cipla and other major companies which are producing antibiotics for human use, and explaining these particular slides that how really antibiotic resistance develops. So before the antibiotics were introduced for the human beings, there were antibiotic resistant bacteria in the population of microbes. But this I explained like this, that when there is a population of dividing microbes, there are always one or two mutants, and these are outnumbered by the ones which are susceptible. But when there is a selective pressure, then all the susceptible ones, they die and the mutants survive. And when this particular antibiotic is on, the mutants continue to grow, and this becomes one of the greatest threat of the human civilization. So there are bad bugs now, which we call superbugs, and there are no drugs for it. Now, what is bad bug? The bad bug is that microbe or the pathogen which cannot be controlled by any of the antibiotics that are present on the shelf. And interesting, the last resort for the medical persons are the carbamine antibiotic. For example, meropenem or imipenem. These are the drugs which are for the last choice drugs. And if that is also being thwarted by the bacteria, then the patient succumbs and dies due to the infection. What is microbiome? There is a term biome fused in it. Means when you talk about the biome, means the biotic and antibiotic factors are to the given environment. So microbiome is the entire habitat. That means of microorganisms, which includes bacteria, archaea, lower and higher eukaryotes and viruses. Not only those bacteria, their genomes and the surrounding environmental condition all constitutes microbiome. So if you remember my talk's title, it is the impact on environmental microbiome. So the entire microbes of the environment, their genomes, right? And the number of microbes which we cannot even cultivate all contributes to this microbiome, right? And these are the major classes of antimicrobial agents and their representative substances. So if you put all the classes of antibiotics till today, aminoglycosides, beta-lactam lipopeptides, macrolides, fluoroquinolones, sulfamides, and glycans. And here in the next, you get the representative substances, right? Now, if you look at this picture, and if you have contracted, UTI, for example, urinary tract infection, so your urine sample is being cultured, and let's say these are the small white paper discs which contains a desired amount of antibiotics, and these are different antibiotics plated on a plate. And you will be finding that there is a zone of inhibition. So this is the halo zone, means the bacteria of your urine, right, is being inhibited in presence of that particular antibiotic. But if you look here, this disc and this disc, you have practically visible no inhibition zone. Why? Because bacteria of your urine, right, that has got resistant to these two antibiotics. And then visualizing this particular picture and the result, your clinician then proposes the drug, he prescribes the drug, and you get rid of UTI. Now, this is the schematic flow of antibiotic resistance carrying bacteria. As you have already seen that how these antibiotic resistance bacteria are getting populated in the environment. So Basically, if you look at these round circles, these are the hot spots where you find all these resistant bacteria and the genes. And one of the important hotspot is the hospitals. From the hospital to the general population, there is a transfer of these bacteria and genes. And these are cattle farms, where from, which is, I told you that prophylactic usages of antibiotic to these farm animals also cause lot of bacteria to become resistant, and they are transferred to as the manure, and ultimately this manure is used in the agriculture field, so ultimately it comes to the environment. And from the hospitals, the entire waste, they are treated in a wastewater treatment plants. And in the wastewater treatment plants also, there are several vectors, for example, air, water, etc., which are given, shown in blue stars. They ultimately carry everything to the environment. And from the environment, there's another route going back to the human population. So this schematic flow of antibiotic resistance bacteria, antibiotic resistance genes give you an idea that
Sir,
antibiotics
sir.
are
Please, sir.
carried to the environment. Yeah?
Sir,
Yeah?
sir, I have to
Yeah.
say something. Sir, as the slides are not visible, so I requesting you to just on your video and without slide, please give your lecture. The slides are not visible, sir. That's why I'm requesting you that please
So,
on your video.
What are you requesting? Yeah?
No, sir. The slides are not visible still now.
These slides are not
So-
visible.
No, sir. That's why I'm requesting you that without slide, please
Sure.
give your lecture and please put on, or please on your video. Actually, unmute your video.
Just hold it. কিছুতেই স্লাইড দেখতে পাচ্ছি না, বলছে আনমিউট করো ভিডিওটা, আমি কিন্তু বলবো না।
কিছু দেখতে পাচ্ছি না।
কিছু দেখতে পাচ্ছি না। তাহলে আমার এখানে ভিডিও Hello, can you speak?
Yes, sir. You are audible.
Can you see the screen?
No, sir.
You're not
Still,
getting the screen, right? Okay.
no, sir.
Still
Still,
not.
it is invisible.
Okay. All right. Let's see. It unmute করতে দাও।
Please turn on your camera.
Yeah, but this camera is not getting open. Turn on.
হচ্ছে না।
Are you getting?
No, sir. Still, it is invisible.
Video getting open from this. It's okay. Hmm.
Sir, click on the entire screen. In the present now button, click on the entire screen. Okay, now it is visible. Okay, sir, now it is visible now.
So how I have to move to the next slide. Yeah. It's visible, right?
Yes, it is visible,
Hello?
but the your audio quality is very poor.
Audio quality is poor. Okay, but can you hear me? How would I tell this? Is it remove their microphone? Will help because the bandwidth. So mute their mic. Increase the bandwidth. So can you hear?
Sir, please turn on your microphone. The microphone is.
Sir, please unmute your audio. Okay, sir. Now
Yeah, can you hear me?
Yes,
Can
sir.
you hear me now?
Yes, sir.
Yes, sir, it is audible now.
It is
Okay,
audible
okay.
And the slides are also visible.
Thank you so much. I'm sorry because there has been a lot of problem. Okay, so let me start and I need to finish it within time and there is a problem with your thing. So we are talking about the resistance. And if you look about the drugs that are available, right? You see, 1, 2, 3, 4, 5, 6, 7, 8 are the different types of targets that the antibiotic do. And they are basically targeting the macromolecular machinery. For example, if it is targeting the DNA replication, then the number one is that type of drug that is going to do that. For example, ciprofloxacin. Number two is going to stop the mRNA transcription from DNA. For example, rifampicin will do that. So 1, 2, 3, 4, 5, 6, 7, 8 are the different targets, right? And the antibiotic classes that we have already seen, for example, penicillin and cephalosporin, they are going to prevent the bacteria from making the cell walls. Sulfonamides and fluoroquinolones, they are going to inhibit the folate synthesis and inhibit the drug DNA synthesis. Tetracycline, aminoglycoside, macrolise, they are going to inhibit the protein synthesis. And how the bacteria is going to get rid of the antibiotics? There are 3 different mechanisms. They are going to make the target resistant to the antibiotic. So the target, if the target is DNA guidase for ciprofloxacin, if the DNA guidase is mutated, then the ciprofloxacin misses to hit the target. Number 2 is there could be several enzymes by which the bacteria can degrade the antibiotic. And the third important is that the antibiotic, when it moves inside the body of the bacteria, it pushes it back to the environment by the use of efflux pumps. And This is the non-target bacterial diversity which is affected by antibiotics. I talked about the farm animals getting feeded with huge amount of tetracyclines to have a good amount of meat as well as milk from these farm animals. You always feed them with antibiotics. So this is a nasty use of antibiotics in farming. And therefore, the non-target bacterial diversity, they get affected by antibiotics. So there are different sources of antibiotic contamination to soil and water. From the household, whatever the disposed antibiotics are then thrown to the community wastewater and ultimately to the rivers and the soil. There are a lot of antibiotics used in agriculture too, in aquaculture, poultry, hospitals, then wastewater treatment plants. These are all the antibiotic sources from which the residues will seep into the soil and water, right? And this is another way of looking into the antibiotic exposure pathway. Means antibiotics used in aquaculture, domestic waste, animal farms, hospital waste, all they are going to pump the residues ultimately to the lakes, streams, and rivers and soil. So a big part of the environment, right, that is soil and water, gets contaminated with antibiotic residues. So in recent years, the scientific researchers have identified additional cause of these antibiotic microbial resistance. What are those? Those are the plants that manufacture antibiotics. And they are causing a lot of problem in China and India. So look at this polluted pond at Bolaram Industrial Area, Hyderabad, where large amount of antibiotics are produced and the wastes are being thrown in a pond. Likewise, if you look at the river Musi in Hyderabad, where there is a major pharmaceutical manufacturing hub, and all these effluents, they are being put into the Musi River. Likewise, the polluted flowing river, that is the water coming from a place, and nearby is the Aurobindo's Unit 5, which is located, some few hundred meters from Isnapur Lake, just inside the Patancheru industrial area. If you look at this, you will be finding that one of the principal pharmaceutical plant of China, you see the effluent pipe, how it is getting delivered into the water sources. If you look at the close-up chemical pools in the lake bed of Kasipulli Lake, Hyderabad, you will find patches of such high degree concentration of antibiotic residues. This is the close-up of an effluent treatment at Godhappa Forum, a village in the outskirts of, Hyderabad. Likewise, I will show you certain CCTV screenshots from China. See, this is one of the effluents coming out. It's flowing into the environment from a pharmaceutical farm in China. So this is the pharmaceutical industry in China, which is just disposing the antibiotic residues into the environment. Similarly, huge tankers filled up of antibiotic residue waste are being delivered in the environment. So what is the ultimate fate of this? Whenever there is a huge amount of such residues in the environment, there is a quantum improvement or increment in antibiotic resistant bacteria and genes in the
interaction, sir. Sir, the slide is blinking again and again.
Well, because it is being supported by Geo, and the entire the main support has lost, because yesterday night there was a great thunderstorm and the entire internet link has failed. So I am pushing this with a Geo thing. Okay, so please bear with it. It is blinking, I know it, because it is also blinking in front of me. So there are major methods of detecting these antibiotic resistance pathogens and genes. So there are laboratory culture method, and you can go for the whole genome sequencing, and you can go for the quantitative PCR for identifying which of the genes that are present in an environment. And of course, you can go for the metagenomics for fishing out the genes that are present in an environment. And if you look at the percentage of resistance to various antibiotics among only 4 gram-negative bacteria which are isolated from blood culture, if you find, Acinetobacter baumannii, this is a dreaded pathogen in the hospital setup. Maybe this is not a big pathogen, but this is a bystander which is going to take over your immune system, right? When such immunosuppressive drugs are delivered to the patients undergoing operations may succumb to death because of the huge infection of Acinetobacter baumannii. And when we have seen these Acinetobacter baumannii coming into the bloodstream, the type of resistance by Acinetobacter, if you look at the ciprofloxacin, just an example, 84% of these blood culture Acinetobacters are resistant to ciprofloxacin, right? So this is the last drug of resort that is left to the clinicians for treatment, and those are the carbapenems, as I talked about. Meropenem and imipenem are the last, and that also is getting resisted by the bacteria which are isolated from the blood cultures. For example, E. coli, Klebsiella pneumoniae, this is a pneumonia causing, and when multi-organ failure is caused by this type of pneumonia infection, means it is moving to the organs via the blood, and 56.6% of Klebsiella pneumoniae are resistant to carbamazepine. So this is a very dreaded situation. If you look at the livestock situations and various studies in India, and just talking about the only one single organism called Staphylococcus aureus, which you got plenty in cow and goat, you find lot of resistance coming up in these type of microbes which are associated with the livestock. So even in the milk samples, raw milk samples, you get a lot of E. coli which are resistant to several antibiotics. One of the important thing in this slide is if you look at the ampicillin, at least 100% of all E. coli that were isolated from cattle raw milk are resistant to ampicillin. So therefore, ampicillin will never work on these cattle, right? So pig and all others, people have tried. So this is the different aquacultures in various studies in India, where Enterobacteriaceae strains, which are isolated from, let's say, tilapia fish, where 42% of those isolates are extended spectrum beta-lactam. They are producers because they are producing beta-lactamases, which is going to take care of all antibiotics of those groups. So tilapia fish is a host of all these bacteria which are producing extended spectrum beta-lactamases. So all these, treatment plants, sewage treatment plants, sewages are coming from the domestic or hospitals. So if you move from the domestic to the hospitals, and if you take only one type of bacteria, you'll be finding that when you are receiving the domestic waste as well as the hospital waste, it has increased to 70% from 25% STP. So sewage treatment plant only receiving the domestic waste containing 25% E. coli resistant to third generation cephalosporin. What is third generation cephalosporin? You know about cefixime, all these drugs, cefuroxime, these are all third generation cephalosporin. And these are resistant to them. So when the STP is receiving only hospital waste, 95% of the bacteria coming out from the hospitals and being treated in the sewage treatment plants are resistant to cephalosporin. If you look at the river, for example, Kaveri, Jamuna, and the rivers from the 5 states, these are the different types of work that the people have done where they have seen the organisms' resistance to different antibiotics. This is also some of the others, Ganga, Jamuna, Kaveri, and Mutha. And if you look at this picture, these are the genes. NDM1. NDM1 means New Delhi metallo-beta-lactamases. So the bacteria which are having this metallo-beta-lactamases, they are going to resist carbapenem. So this is a dangerous type of antibiotic resistance genes that the microbes or the bacteria are hosting in order to thwart carbapenems. So carbapenemase has entered into the rivers. Now let's see what about West Bengal. So you see, for the last 20 years, I've been working on this problem. So it is also a journey from where we started. So this is a beautiful picture of Siliguri with 2 bridges, and underneath flows the Mahananda River. And we have 3 sampling stations across this. SS2 is the one which is flowing through the heart of the city. SS1 is the upstream. SS3 is the downstream. And this is the SS2, where you find lot of cattle and others and all waste. They are delivered in the waters of Mahananda. Another city of northern part, which is we call as a twin city, Siliguri and Jalpaiguri. Jalpaiguri is a mighty river, Tista, in the outskirts, and in the middle of the, which is bisecting the Jalpaiguri city, is the Kerala River. So this is one of the Kerala River's upstream stream that you are visualizing. And these are the methods that we have used. So all these methods, whole genome sequencing to laboratory culture, to metagenomics, to qPCR, all are being practiced in my laboratory for the last few years. And this is the type of design that we have to understand and find out the abundance of these metallo-beta-lactamase possessing strains in the rivers, means the Mahananda and Kerala River. So these are the sites, sampling sites that you have seen. So we call them as upper, middle, and downstreams of both the river. And when we plot the data, you will be surprised, the upstream data has a lesser amount of inipinem resistance. But if you see the midstream, which actually carries a lot of hospital wastes, they have a huge abundance of carbapenem-resistant bacteria. And same too in case of Kerala. And the picture is similar to that of Mahananda. And I'm sure if you do it in any cities across West Bengal, you will find the same picture. And when we have seen this, see, there are several genera which is possessing NDM1. And this is one of the dreaded genes by which the bacteria is resistant to the most advanced antibiotic. So you have Pseudomonas, Acinetobacter, Proteus, Escherichia coli, all isolated from the rivers of Mahananda. And these are giving you the type of gene cassettes they hold, the type of alleles they have, and we have confirmed all these things. So now let's enter into the half-life of these antimicrobials in the surface of water. So if you look here, the norfloxacin having 77 days half-life. So when it is being excreted in the environment, means after taking, and most of the drugs are not metabolized. They come out in the excreta and go into the sewage system. And therefore, this surface water half-life is very important for considering something. So antibiotic resistance genes are highly abundant in the wastewater and environmental samples. So this is giving you the beta-lactam antibacterial genes. For example, BLA genes means beta-lactamases of different types. They are most abundant in the WWTPs, means wastewater treatment plants, river water, and there are groundwater also. There are also no reports still yet from these type of antimicrobials we have. So this is the case of tetracyclines. So abundant TET genes are available in the river water as well as in the wastewater. Abundant genes responsible for resistance to macrolides, quinolones, sulfonamides, and also other substances that we find in the wastewater and environmental samples. So animal farm, human therapy, and the produce where it is being produced, that is the pharmaceutical industries, all are discarding the waste, the antibiotic waste into the system. Ultimately, that goes to the soil, and that leads to the spread of these ARGs via horizontal gene transfer. The impact is long term. The soil microbial community responses, the microbial community is devastated. They are getting all these genes, and ultimately these genes are getting widespread in the environmental microbiota. So there is a transfer of antibiotic resistance in the soil, and there's a high risk of animal and human infection out of this transfer of antibiotic resistance. And this is the maximum residue limits that one can have in veterinary residues. But we are getting the residues much above what is being maximum residue limits that has been provided by the veterinarians. So these are the possible paths that I have explained earlier. And this is one of the paper that came out in Chemosphere, which talked about the persistence of broad range of antibiotics during calf, pig, and broiler manure storage. So how they have experimented, you see, this is about the pig manure, this is the broiler manure, this is the calf manure, from where they have set up the persistence study, means how long the antibiotic residue stays within the manure. And ultimately, they have statistically validated all their findings, and they have come to this calculation that tetracycline, which is heavily used in farms, okay, DT means the dissipation time. That means,
Sir,
yeah.
I have to say something. So as there is a scarcity of time, please conclude it early, sir.
So let me end it, right?
Not like that, sir, as the time is very limited.
No, but I understand. I understand. So I believe I need to come to a conclusion, right? So what is the conclusion? Many factors that control the biodegradability of contaminant in the environment. And with regard to all these type of antibiotic residues, right? And we need to attempt the technology, and before using the technology, you need to understand the catabolic pathways. And there are several, the work on the bioremediation has just started. So contaminants must be bioavailable and is optimal condition. So people are trying to understand the environmental conditions that are required to promote the growth of this desirable organism, and then engineer these environmental conditions needed for this, bioremediation. So thank you for listening and interrupted deliberation from my part. I'm sorry to the listeners. And this is what I can acknowledge is the group that has, contributed to a 20 years long study. Thank you very much.
Thank you, sir. Thanks for sharing your valuable words. Maybe there was some technical error, but still it was quite knowledge enriching. Thank you, sir. Now, the next resource person is Dr. Bernard F. Rodrigues, Senior Professor in the Botany Department at the Goa University. Professor B.F. Rodrigues has been working on AM fungi and reclamation of iron ore mine wastelands, and he has 30 years of teaching and research experience. His 135 research papers in different national and international journals got published till date, and 12 students have completed PhD under his supervision, and 5 are pursuing their doctorate. Now, please, sir, we are waiting for your kind deliberation.
Yeah. Hello, am I audible?
Yes, sir, you are audible.
Okay. Good afternoon, everybody. The respected principal, Madam Mohanta, then all the honored guests, friends. Well, at the outset, let me thank the organizers for giving me this privilege to make this presentation. We all know that our environment needs protection. We have to give top priority. And in this connection, we need to know that we have borrowed the planet from our forefathers and we have to use it sustainably and then give it to our coming generations in the same way we have used. However, we have seen that there are a lot of environmental issues because the way we have handled the environment has been bad and for which we are suffering today. Well, presentation is here. Put it on. Well, I'll be talking on the
Yes, sir, the slide is visible.
Yeah, I'll be talking on the revegetation of mine lands using mycorrhizal fungi. So my presentation has been divided into 4 parts. The first part would deal with the iron ore mines and their characteristics. The second part would deal with the various strategies for reclamation of the iron ore mine lands. The third part would talk on the mycorrhizal fungi and the production of inoculum. And finally, the fourth part would talk about the role of mycorrhizal fungi in the reclamation of iron ore mine lands. Well, the state of Goa is a major producer and exporter of iron ore in the country. Since inception of mining, more than 300 tons of iron ore has been exported. Major minerals manganese and bauxite. The estimated reserves of iron ore are around 400 million tons and are expected to last for another 25 to 30 years at the present rate of mining. In fact, mining of iron ore started way back in the early 1950s in Goa, where the bullock carts were used for transporting the ore. In Goa, basically, we have open cast mines, wherein before mining, the mining operation, the vegetation cover has to be removed, followed by the removal of the soil until the ore is reached. But the mining operations produce 2 classes of waste, namely the piles of overburden, waste rock and lean ore, which constitutes the reject dumps. These reject dumps reach to a height of about 50 to 60 meters. Whereas the second class of waste termed as tailing is a fine-grained waste resulting from the ore beneficiation process and deposited in large man-made basins. They are called as the tailing ponds. You can see the tailing ponds here. So the tailing ponds, they dry up in summer, giving such an picture. Now coming to the benefits of mining, some of the benefits include it generates employment, which is very important. It helps foreign exchange earning, and mining is a basis for all other industries, and therefore no progress is possible without mineral development. However, there are numerous harmful effects of mining. This includes mining causes major disturbance to the landscape. There is destruction of forest areas. Since 1961, 50,000 hectares of forest land has been destroyed due to mining activity. Mining generates enormous amount of waste. Since 1961, again 900 to 1,000 million tons of waste has been generated. Mining drastically affects the agricultural land. Mine rejects being unconsolidated are prone to slumps and slides due to heavy monsoon rain. In Goa, mining has killed the fertility of over 10,000 hectares of agricultural land. Besides, mining causes noise pollution, the blasting operations, movement of heavy vehicles, operation of heavy machinery leads to noise pollution. It also causes dust pollution. Operations such as blasting, crushing, and transportation cause dust pollution. The dust has demanding effect on vegetation by blocking plant pores. It reduces light penetration and hampers photosynthesis. Now, the objectives of this work were, one was to achieve a self-sustaining ecosystem, to accelerate the rate of natural succession, and to increase the biodiversity, thereby bringing successful rehabilitation of the once degraded area. Now, to begin with, we started with the survey studies. We carried out detailed survey studies of the vegetation cover that appeared on the fairly established iron ore mine sites. The purpose of this study was to get a clue on the type of plant species that appeared in mining. We also checked the growth performance of these species. It was seen that a number of plant species appeared at these sites, at the fairly established sites. Most of these species showed stunted growth with woody stems. However, there were also plant species like this, which showed better growth, indicating that this species could possibly be utilized for revegetation of the degraded mine sites. Here also we have Callicopteris and Tremorentalis. It was observed that on the steep slopes, the mosses and pteridophytes had established, thereby checking soil erosion. However, it surely must have taken decades for the establishment on these slopes. So, faster and better methods are now available to check and minimize the erosion of the dump material. This includes terracing, then growing of grass species. There's the short grasses because the tall grasses are prone to fire. Then plantation or planting legume species on the slopes that can rapidly cover the surface and also enrich the soil with nitrogen. Besides, tree species such as Anacardium occidentale, commonly known as kaju, they are found to be excellent in checking the soil erosion. So we studied the species diversity with the age of the mine versus the number of species. It was observed that the older mine sites had greater plant diversity compared to the recently degraded mine sites. A 10-year-old mine site recorded 29 species, whereas a 50-year-old mine site recorded 250 species. The study showed that herbs dominate as pioneer species and appear soon after the mining activity. It was also observed that the members of Leguminosae, Asteraceae, and Poaceae families dominate the recently degraded mine sites. However, their number gradually decreases in the older mine sites, indicating the crucial role that they play in the initial stages of revegetation process. We also carried out preliminary polyboic bag experiments to compare the growth of a number of plant species grown separately in mine soil and garden soil. The results of our study revealed that most of the plant species showed poor growth in the reject material than in the garden soil. However, there were few plant species which showed growth comparable to that in the garden soil, indicating that these are potential candidates for the revegetation program of the mine lands. Later, we carried out analysis of the mine rejects. Based on the analysis, it can be concluded that the mine rejects have high clay content, the results which results in compaction, greater runoff, and poor aeration, thereby affecting the plant growth. The mine reject also showed acute deficiencies of macro and micronutrients that results in poor plant growth and survival. Also, a number of deficiency symptoms were observed in these plants that were growing on the mine sites. The mine showed reject showed low electrical conductivity, indicating that there is no likelihood of any salinity problems. There were extremes of water and temperature. In summer, the temperature increased, and mines being brownish in color, the temperature in these areas was higher than the surrounding temperature. And there was deficiency in the organic matter. Now, since these are materials from 50 to 60 meters depth, there was hardly any organic matter, organic microbial activity, so there was no organic matter. And the pH was neither highly acidic nor alkaline, indicating that the plants would survive at these sites. Now, coming to another observation that we made is that the mining companies, after mining, they would plant a handful of species. So they would carry out an artificial plantation by using fast-growing exotic species such as Acacia auriculiformis, Casuarina acquisitifolia, and eucalyptus species, which requires increased attention after planting. Now, these monoculture plantations are prone to dangers of pests and diseases, and these pests and diseases can wipe out the plantations in large areas of the revegetated mine lands. In short, there's hardly any plant diversity in such areas. Also, no native plant species are used for revegetation of such areas. Again, the ground cover is bare minimum, which has only these PD species. Therefore, to bring maximum diversity, we divided plant species into several categories, such as the naturally colonizing plant species, the native plant species, the grasses, herbs, shrubs, legumes, trees, and exotic plant species. So we prepared this flow chart, which gives the different strategies for artificial revegetation of the land, following the land degradation due to mining, which would assist the reclamation process. It is well known that most of the microbial activity is in the first 15 centimeters of the soil. So, collecting topsoil before the mining activity and storing it and using it in a given span of time would help the revegetation process. Besides, the microorganisms such as bacteria, rhizobium, the fungi like the mycorrhizae could be used. I also already talked about the selection of plant species. The terrace mine dumping, where terracing is done, this is also talked about. And then the microorganisms like earthworms, termites, and ants would play an important role in the fairly established mine sites. The organic manure such as sewage sludge, farmyard manure, seaweeds, green manure would help. And then reaping the surface to overcome compaction, especially where there is a lot of movement of machinery, would help for rapid natural succession. So that once an area that was a forest, there's deforestation leading to mining, degraded land. By these the methods of artificial revegetation, we can increase the pace for natural succession. So besides selection of plant species, is a critical factor. Fast-growing native plant species would ensure higher success rate. We recommend the use of the two plant species, namely Trema orientalis and Macaranga peltata, besides others. Being pioneer species, these two are fast-growing and are therefore suitable candidates for initial revegetation of the degraded mine lands. The main purpose I mean, the main problem that we faced with these two species is that although these species produce millions of plants, but they show very poor germination, which is less than 0.01%. And this germination is non-synchronous, and therefore raising them in large numbers in nursery was an issue. So we developed effective protocols through seed germination, the seed treatment, by using tissue culture techniques as well as the physical methods to overcome this problem. Here you can see that in case of Makaranga peltata, we have used the seeds. The seeds show viability, but the seeds do not germinate. However, we could break the seed coat artificially by using this equipment, and there was germination. So also we have used tissue culture for both raising both this is Stemma orientalis plantlets and Makaranga peltata plants, so that we could produce large number of plantlets and then they could be used for the revegetation purpose. So we increased the germination percentage to about 85%, from that 0.01% to 85% in both the species, and we could achieve synchronous germination, and therefore we were successful in raising them in nurseries. So this is a nursery where we have used these plant species, both the plant species, which are important. They are fast-growing species. Now coming to the third part, we talk about mycorrhizae and how we have used these mycorrhizae for reclamation of the mainlands. So mycorrhizae, we all know that these are fungi. It's a symbiotic association where they play a very important role in plant nutrition and plant growth. So these fungi are obligate symbionts, symbiotically associated with living plant roots. Both the partners, that is the fungus and the host plant, are benefited as there is a bidirectional flux. The plant receives nutrients, especially phosphorus, besides other macro and micronutrients. Phosphorus shows poor mobility in the soil, and this results in phosphorus depletion zone. It also gets readily fixed as phosphates of iron, aluminium in the acidic soils, and calcium in the alkaline soils, depending upon the pH. So the mycelia, being much finer than the roots, can reach the minutest of the crevices in the soil and absorb phosphorus and make it available to the plant. Besides, these fungi provide numerous other benefits to the host plant. So it is basically the plant fitness that is taken care of by this mycorrhiza. The arbuscular mycorrhizal fungi, they are prevalent in the tropics and play an important role in plant nutrition since tropical soils are deficient in available phosphorus. They act as phosphate mobilizers, thereby assisting in improved plant growth and productivity. The association helps to increase the absorptive surface in the mycelia. They go beyond the phosphorus depletion zone, absorb phosphorus, and make it available to the plant. In turn, the mycorrhiza gets carbon source from their host. Of course, there is no host specificity. However, there is host preference, and this host preference or this aspect needs to be exploited so that we can minimize the plant growth and productivity. Presently, more than 260 arbuscular mycorrhizal fungal species have been identified. This identification is based on the spore morphology. The spores, they vary in size, subtending hyphae, presence or absence of septum, sporocarpic, non-sporocarpic forms, etc. You can see in the slides the species belong to the different genera. This belongs to Echinospora. You can see the size, shape, the color, the ornamentation, they vary based on which they have been identified. So I'll quickly go through these slides. These are some of the sporocarpic forms. So based on these characteristics, the species, AM species are identified. Also, there are structures by which we can identify these, that the plant is colonized by these AM fungi. They produce these peculiar or characteristic structures like vesicles, which store lipids and they act as chlamydospores. These are the intraradical and extraradical hyphae, which are non-septate. Very rarely they are septate, and they produce these intraradical and extraradical spores. Besides, they produce the orbicals. Orbicals are sites for transfer of nutrients from the plant to the fungus and fungus to the plant. And these are the auxiliary cells which are found in some of the genera. So these are features whereby we can identify and say that the plant is mycorrhizal or not. So coming to AM fungal inoculum production, there are different ways of producing the inoculum, like the pot cultures, aeroponics, hydroponics, and monogenic cultures. So we'll concentrate on the preparation of pot cultures that we have been working with, and we have been working with the in vitro culture, that's the monogenic culture of the culture methods. So for that, we have to produce first we have to trap the spores, of course, I'll not go into the details of that. And then we have to develop monospecific cultures. So these are spores of a single species, which are being maintained in the polyhouse. And which is dedicated to mycorrhizal inoculum. The species, different species are kept here. And you can see here that we get very clear and clean spores since they are in this condition. The spores are clear and clean, and they are not parasitized as we get in the rhizosphere soil. So these spores can be used for in vitro germination. So pot culturing is one method. Other method is the organic cultures, whereby the spores, the clean spores, are disinfected and they are cultured. They are germinated in the petri plates, and after germination, we can get thousands of spores in the petri plate. They multiply, and these spores could be used as inoculum. So again, you can see the young spores which are highly viable, which could be used for preparation of the inoculum. So we have used these are some of the slides where we have used the pot culture inoculum, multiplied them in the pots, and used them for different species have been used against control, and we can see the definite increase in plant growth. This is in case of cashew and some terminalia species. This is in case of Trema orientalis. We have used the different amendments, including farmyard manure and vermicompost, and we found that when they are used in combination with mycorrhiza, it gives a better effect, better growth. In case of Macaranga peltata also, you can see here the growth that is in garden soil, and the mine soil is much less as compared to these where we have made the amendments. So that this is in the mine spoil, indicating that the mine spoils are deficient in nutrients, therefore the plant growth is poor. Whereas here, after adding the amendments, suitable amendments, we could increase the plant growth. This is in case of cashew, where we have used the in vitro culture. So in vitro, that is prepared culture or monogenic cultures which are prepared, they were used for this particular experiment. And you can see the control sterilized sand, and this is in case of control unsterilized sand. You can distinctly see the difference in growth and performance of the cashew plants. And then when the roots of these plants were checked for colonization, we could get the colonization. This colonization is a characteristic feature of is a mycorrhizal colonization by AM fungi. You can see the vesicles here, clear vesicles. You can see the extra radical and intra radical spores, indicating that the plants were colonized and that the effects were because of the mycorrhizal colonization. We also work with the ectomycorrhizal fungus, Pisolitus albus, where the fruiting bodies which are found in nature under the Acacia auriculiformis plants, just at the beginning of monsoon, which we collected the young fruiting bodies, and they were grown in culture, and we could develop the inoculum. So within a month's time, the entire petri plate was full with the inoculum, and this was also used as the inoculum. The tissue cultured plants of Trema orientalis and Macaranga peltata, they were grown, and we added the mycorrhizal inoculum so that they were mycorrhizal, and these mycorrhizal plants do better in the mine soil than the non-mycorrhizal plants. We also used the mycorrhizal inoculum for raising the saplings in the nursery. So a large number of plants or plant species were raised in the nursery by using mycorrhizal inoculum. And then later on, we had to grow these saplings on the mine sites. So at the mining site, these saplings were grown, and then they survived. We also have results where the mycorrhizal plants did very well compared to the non-mycorrhizal plants. Here you can see how the vegetation has come up. So the entire dump has become green, has been revegetated, and as these plants grow, it would be greener. And then a close-up of some of these plant species. This is Carissa carandas, where even in summer, when even the grasses have dried, the plant is surviving. This is another Syzygium cumini, or the jamul plant. This is Calicopteris floribunda. So, large number of plant species we planted. We did other experiments by using a grass legume mix, where we had broadcasted the seeds of grass and legume around these plants, 1 meter around these plants, and they have given better results. So, we by doing all these activities, we developed a self-sustaining ecosystem where not only plants but also other organisms, the birds and the animals, the microorganisms like the termites, and they are also very important in the degradation part. So they would visit, and the whole system becomes, or the area becomes self-sustaining. There was increased diversity in the fauna, like all these, like the mammals and birds and reptiles, and their number increased. And this is one area that we see. Of course, this is not our work, this work was done in the 1960s, soon after revegetation, soon after the mining activity. The area was covered with the native plant species. This was an experiment done some 70 years back. And you find that except for these two patches, the area looks completely green. It has rich climax vegetation, and nobody would say that this area was once a barren mine dump because of mining. Therefore, it is true that in the process of mining mineral resources from the earth, disturbance to environment and ecosystem is unavoidable. In other words, mining is to some extent an unavoidable destructive process. It is true that there are several problems in mine waste in terms of erosion, environmental pollution, damage to adjoining agricultural fields, forests, etc. However, these hazards appear to be within measurable limits and can be easily ameliorated to a significant extent by extensive research and planning to control the impacts. Well, finally, I would say that the mining industry need not lead to degradation of environment if a combination of imagination, care, and scientific skill is applied by those who are involved in such programs. Thank you.
Thank you, sir. Thanks for accepting our invitation and sharing your valuable thoughts with us. Now the next session is for question and answering, and this session will be run by Dr. Navanita Chattopadhyay, SACT, Department of Botany, Chagda College.
Good afternoon, everybody. We have got some interesting questions from the participants. I request the experts in the panel to kindly respond.
Okay, please.
Tritiman, question is, can we go for sewage treatment by the use of beta-lactam antibiotics? If yes, please give the example of such antibiotic.
Now, one thing is that when sewage is contaminated with antibiotics, you cannot treat with the antibiotic. Okay, so you need to treat with the antibiotic degrading bacteria. So therefore, the technology is the waste treatment plants must be fortified with antibiotic degrading bacteria. And there are examples that bacteria can consume penicillin. And these are all coming up in the last 2.5 years. Okay, so it is known that penicillin can be consumed as substrate. So if some bacteria use it as a carbon source, and if you have those bacteria in the sewage treatment plants, then the penicillin residues will go off. Next question, please.
Okay, sir. Chitra's question is, can these microbes be useful for small-scale treatments with limited resources?
Yeah, but the research is not that level has developed where we get the ready technology to do this at a small scale. So therefore, if you look at the picture of India's perspective, bioremediation study with respect to antibiotic residues practically negligible. Only few theoretical works have been done. And one of the breakthrough has come out from our laboratory that certain NDM1 bacteria, means beta-lactamase, particularly metallo-beta-lactamase containing bacteria, can be engineered to get rid of the beta-lactam residues in the environment, for which we have worked out the pathway. Means this has to enter into the central metabolic pathway, and that happens via phenylacetic pathway. So the entire pathway was deciphered. Now we are in a process of engineering that bacteria for doing small scale studies for getting rid of beta-lactam residues in the environment. Thank you. Next question.
Okay, sir. Next question is, how long do mycorrhizal fungi survive in soil without an active root system?
I mean, Professor Rodriguez.
See, the mycorrhizal spores can survive for about 2 years. They are viable. It depends upon the predators. If there are more predators in the soil, then they can be, they get damaged, they get bruised, and as a result, they can lose the viability. But for 2 years, as a soil inoculum, one can store it.
Okay, sir. How long do commercial mycorrhizal inoculums survive?
For 2 years, same as.
Okay, sir. Thank you. Now we have come almost at the concluding part of the webinar. I request Dr. Anjan Sengupta to express vote of thanks.
Hello, am I audible? Hello.
Yes, audible.
Okay, audible.
Audible.
Good morning.
Yes, sir, you are audible.
Good afternoon, everybody. On behalf of the Department of Botany, Chakda College, give it a great pleasure to propose the vote of thanks to all who have helped us in making the webinar a resounding success. First and foremost, I thank our chief guest, Honorable Vice Chancellor, Dr. Shankar Kumar Ghosh, who despite his busy schedule, has found time to praise the occasion. We are grateful to respected Principal Ma'am, Dr. Shagota Das Mohanto, for her constant support and encouragement. My heartfelt thanks to the guest of honor, Dr. Chandan Sengupta, ex-professor of Botany, University of Kolani, for delivering the keynote address. I must express a deep sense of appreciation to the honorable resource person, Professor Dr. Ranadhi Chakraborty, a renowned microbiologist and biotechnologist, for his insightful speech on antibiotic residues in the environment and the ways to its mitigation. An eminent researcher and teacher, Professor Dr. B.F. Rodriguez, has been working on mycorrhiza for the last 30 years. We are greatly indebted to you, sir, for your thought-provoking speech on revegetation of mining areas with mycorrhizal fungi. I would like to thank Dr. A. Roy Chaudhary, head of the Department of Botany, for making the event a grand success. I also extend my thanks to Dr. A. K. Nandi, convener IQSC, Dr. N. Chattopadhyay, deputy convener of the webinar, Dr. A. Banerjee, program host, and Dr. M. Devnath, program host. and all the faculties and staff of the Department of Botany for their untiring effort in pulling off the event. My sincere thanks to all the participants from various corners of India and abroad. We appreciate your patient cooperation all throughout the session. We have been fortunate enough to have a team of motivated and dedicated faculties and staff members of Shakta College who have made this event a great success. Thank you all. Here is an important announcement. E-certificates would be awarded within 10 days to the participants who have registered themselves as well as filled up the feedback forms. The feedback link would be provided on the respective WhatsApp group, and the link will remain open till tomorrow 11 a.m. IST. Now, over to Dr. N. Chattopadhyay.
Now we are concluding this effective and informative webinar, and I declare the webinar closed. Thank you.